The human immunodeficiency virus, or HIV, stands as a formidable threat to global health. This virus specifically targets the immune system, the body's intricate defense network responsible for warding off infections and diseases. HIV is transmitted through bodily fluids, primarily through unprotected sexual contact, sharing needles, or from mother to child during pregnancy, childbirth, or breastfeeding. Understanding how HIV spreads is crucial for prevention. Left untreated, HIV can progress to acquired immunodeficiency syndrome, AIDS, a late stage of infection marked by severe immune system damage. HIV's impact on the immune system is profound. To grasp the gravity of this virus, we must first understand the crucial role of CD4 T cells, the very cells HIV targets. CD4 T cells, also known as helper T cells, are white blood cells that play a pivotal role in the immune system. These cells act as sentinels, constantly patrolling the body for signs of infection. When a CD4 T cell encounters a foreign invader, such as a virus or bacteria, it recognizes the threat and springs into action. The CD4 T cell's primary function is to activate other immune cells, such as B cells and cytotoxic T cells. B cells are responsible for producing antibodies, specialized proteins that bind to and neutralize pathogens. Cytotoxic T cells, on the other hand, directly destroy infected cells, preventing the spread of infection. The activation of these immune cells is a highly coordinated process, with CD4 T cells acting as the orchestrators of the immune response. They release signaling molecules called cytokines that direct other immune cells to the site of infection and instruct them on how to combat the threat. Without CD4 T cells, this intricate communication network breaks down, leaving the body vulnerable to attack. HIV infects CD4 T cells by exploiting the very receptors these cells use to recognize and fight pathogens. The virus attaches to specific proteins on the surface of the CD4 T cell, effectively hijacking the cell's machinery for its own replication. Once inside the CD4 T cell, HIV releases its genetic material, RNA, into the cell's cytoplasm. The virus then uses an enzyme called reverse transcriptase to convert its RNA into DNA, which can then be integrated into the host cell's genome. This integration allows the virus to take control of the cell's replication machinery, forcing it to produce more copies of the virus. As the infected CD4 T cell produces more viral particles, these new variants bud off from the cell, often destroying the host cell in the process. These newly formed viruses then go on to infect other CD4 T cells, perpetuating a vicious cycle of infection and cell death. A weakened defense: the consequences of HIV infection. The progressive destruction of CD4 T cells by HIV has devastating consequences for the immune system. As the number of CD4 T cells declines, the body's ability to fight off infections is severely compromised. This leaves individuals with HIV increasingly susceptible to opportunistic infections. Opportunistic infections are caused by pathogens that rarely cause illness in people with healthy immune systems. However, in individuals with weakened immune systems, these pathogens can take hold and cause serious, often life-threatening illnesses. Examples of opportunistic infections common in people with advanced HIV infection include pneumocystis pneumonia, a severe lung infection, and Kaposi's sarcoma, a rare cancer that causes lesions on the skin and internal organs. As HIV infection progresses and the CD4 T cell count continues to fall, the body becomes increasingly vulnerable to these opportunistic infections. This progression from HIV infection to AIDS is marked by a significant decline in CD4 T cell count and the development of one or more AIDS-defining illnesses. The battle continues. Challenges in treating HIV. While significant advancements have been made in treating HIV, challenges remain in eradicating the virus completely. One significant obstacle is the virus's ability to establish latency. During latency, HIV integrates its genetic material into the DNA of host cells, essentially hiding from the immune system and antiretroviral drugs. These latent reservoirs of HIV can persist for years, even in individuals receiving effective antiretroviral therapy. Another challenge in treating HIV is the virus's high mutation rate. HIV replicates rapidly, and during this process, errors can occur in the copying of its genetic material. These mutations can lead to the emergence of drug-resistant strains of HIV, making treatment more difficult. Despite these challenges, ongoing research continues to advance our understanding of HIV and develop new strategies for treatment and prevention. Current antiretroviral therapies are highly effective in suppressing HIV replication and preventing the progression to AIDS. However, ART needs to be taken lifelong and does not eliminate the latent reservoirs of HIV in the body. The quest for a cure, research and hope. The search for an HIV cure and effective vaccine remains a top priority for researchers worldwide. Current areas of research include developing gene editing techniques to remove integrated HIV DNA from host cells, boosting the immune system's ability to recognize and eliminate HIV-infected cells, and developing vaccines that can prevent HIV infection. Understanding how HIV attacks the immune system is crucial for developing effective treatments and prevention strategies. By targeting the virus's ability to infect and destroy CD4 T cells, researchers aim to develop therapies that can control the virus and potentially eradicate it from the body. The global fight against HIV requires a multifaceted approach, encompassing prevention, testing, treatment, and continued research. Public awareness campaigns, access to affordable testing and treatment, and reducing stigma associated with HIV are all essential components of a comprehensive response to the epidemic. By working together, we can strive towards a future free from the threat of HIV.